Hello, good morning, all. This session is about week 10. Okay, we can start. We can start the session. You can also ask the week 11, 12 if you've already gone through. Okay, you feel free to ask questions or write on the chat box. Yes, those who are online, if you have any questions, you can ask, or if there are no questions for this session, we can end the session then. This session was for week 10, where we discussed about single cell genomics, epigenetics, spatial transcriptomics, metagenomics, and their role in human health and disease.
Hello, sir.
Yes.
Okay, but can you please tell about something about the spatial transcriptomics?
Okay. Yeah, you want to ask a question in spatial transcriptomics. So what is the question?
Yes.
What is the question?
Okay, I just want to know about the basic concept of the spatial transcriptomics and what is the positional context in this. I want to know about this.
Okay, so in spatial transcriptomics, okay, so first of all, I will say about transcriptomics, where what you do is you take a tissue, okay, because from a single cell, you may not get that much RNA, and other techniques were also limiting earlier. So you take tissue and which contains multiple cells, and you extract the total RNA. Okay, then go for either type of RNA. It could be mRNA or any regulatory RNA. Okay, like that. Okay, and their sequencing or expression pattern using microarray, right? Now in spatial transcriptomics, we want, and then we can go to single cell, right? Where you can take out that cell and then do the transcriptomics profile of that cell, because in a tissue, the cells may behave differently in terms of gene expression, and also sometimes some cells may be healthy and some are cancerous, or there could be tissue differentiation or heterogeneity. Okay, so that is single cell transcriptomics. Now, spatial transcriptomics wants to capture the expression of a tissue where there are different cells, okay, live. Okay, so basically you can have a tissue from which you can read the transcripts level, their expression, okay, without disturbing the single cell or lysing the tissue. Okay, because earlier it was a problem that when you are taking out the cell from the context of the tissue, it may have undergone some kind of stress or like that. Okay, so basically this is very important for like precision medicine, and also to understand the uh, differential gene expression in context of a tissue.
Okay, it's important for differential DNA construction, right?
No, to understand the differential gene expression.
Gene
This,
expression. Okay, okay. How
so
the gene is
yeah,
expressed in various ways, right?
yes. So in different tissues.
Or in case of the different tissues.
Yes.
Okay, okay. Hello, sir.
Yes.
Okay, I'm just telling about one thing. Our exam will be on 30 April. So, and it should will be conducted in the mode of both yeah, online MCQs and descriptive questions, right?
Uh, so there will be. So you have gone through the assignments, right?
Yes, I assignments. It is all.
So the final exam will be on the same patterns that we have already covered in the assignments, right?
Oh,
The type of questions and pattern will be similar. Okay.
okay, so it will be only a computer-based test, only the MCQ option choosing, right?
Yes. There could be some small, calculations based questions,
Okay,
but,
some calculation-based question, and the calculator will be given on the computer screen.
You may not need a calculator because
It is genomics related,
it
basically
is, yeah,
biology related.
it is simple to calculate on pen and paper, I think. Okay.
Okay,
Okay.
but
Yeah.
okay, so I'm just asking one thing. Beside your study materials, is there any another another materials for the exams are in the NPTEL portal of your course is available or not?
Okay. So exams are mainly based on the areas and lectures that we have covered. Okay. And those should be sufficient. And also based on the general description and discussion that we had during the presentations and lectures. Okay, so then and also based on like general understanding that you should have after going through these lectures, right, and this course. Okay, so it will be based on that. And yeah, and I've also mentioned many times that the reference from which I have taken some knowledge extract or some image or something, right? So those who want to go in detail, they can always refer to those things, right? But mainly it will be from what we have taught in the lectures. And there is a question by Elizabeth Thomas that Elizabeth has just started the research in area of genomics, but Elizabeth is from computer science field. Okay, so is it easy to transit to this area? Also, for same reason, I have joined this course. Okay, so good question. So as a biologist, I feel that uh, for computer science people, it is much easier to venture into biology, right? Because you will be dealing with a particular topic or although understand increasing your knowledge in biology and going through the details may matter in the end. Okay, but uh, it would be like from biology you will be given with a question in, okay, which can be solved computationally. Okay, and for that question you will be given all the information that is needed. Okay, right, so you can solve the question computationally. Okay, so it could be like writing a program or algorithm or a script or making a web server, okay, database, right? So right, and when you have started doing this, if you do several projects, you will definitely learn a lot of biology. Okay, um, okay, but now when I say that about thinking in biology, okay, so that could become a limitation unless you go through the area of biology that you are dealing with. Okay, you can pick a subset, like it could be say transcriptomics or metagenomics, right? Uh, okay, or basically genome sequencing. So that way you can gain knowledge in that area of biology instead of learning the entire biology, okay, which is vast, right? Then you can also start thinking about the problems and opportunities that may exist. Otherwise, you will be mainly collaborating with the biologists to solve their problems and given their inputs. Okay, but for a competent person, it is not difficult to solve the biological problems.
Hello, sir.
Yes, Elizabeth.
Sir, I have just some doubts. What is the difference between transcriptomics and RNA? Is it the same?
Transcriptomics and RNA, right?
Yeah.
Okay. So when you okay, the first word should be transcriptome, right? Okay. And so transcriptome is the total content of all the transcripts, which are actually the RNA, okay, which are made in a cell. Okay. So a transcriptome, when you say I am extracting a transcriptome, or right, so you are extracting the total RNA which is present in a cell at that point of time. Okay, and that total RNA is basically the transcripts that have been formed by from the DNA. Right. So transcriptome is this, and when you study transcriptome using any methodology, or in transcriptome there could be the coding RNA, okay, or structural RNA, like ribosomal RNA, okay. So or non-coding RNA. So those will be, that can be called as transcriptomics. So transcriptomics is basically the study of RNA. Okay.
Okay, so it is the same, like RNA sequence, many times I've seen in papers.
Mm-hmm.
So it is same as transcriptomics, right?
Yes. So sequencing RNA is one part of transcriptomics. Otherwise, you don't need to sequence. You can directly detect RNA, right, using microarrays, and okay. So that is also transcriptomics.
Okay. Sir, also which is the latest field, like in case of, let's say you mentioned about spatial transcriptomics.
Mm-hmm.
So like that single cell, I'm currently working on the single cell analysis.
Mm-hmm.
So is it the latest trend?
See, all have their own significance and applications, right? All basically now, from when we are doing transcriptome, then we start doing single cell transcriptome, although parallel activities are also going on earlier. And then now spatial transcriptomics. So it is technological advantage or advancement that has happened, where you can study you can capture basically the mRNA from the adjacent cells in any tissue section. Okay. And once you have captured that mRNA from the cells, then basically you are reading uh, each cell's mRNA, right? Um, in the context of the tissue. Okay. So the earlier techniques used to sort the cells or separate single cells either by fax or laser microdissection. But here you are not separating the cells, because that may also change their like expression patterns and put additional stress on the cell, and transcriptome changes because of that. So here you are taking the entire tissue and then extracting the mRNA. So okay, now this has happened. In the future, you may expect that, okay, like even you don't have to take the tissue out, you can directly read the expression from any organism and like that. Okay, so these kind of technological advancements will happen. Okay, and these are all based on the new technology or equipment that companies offer, and based on the demand and requirement also, right? But the underlying principle, or the method of analysis, okay, and your understanding about transcriptome and the area will be like what we have gone through in the transcriptome or single cell genomics and like. Okay. So the biology will remain the same. It is only that how effectively now we are able to read that information in the context of a tissue, cell, or an organism.
Okay, sir. So then we were searching for multi-omic data.
Mm-hmm.
So like what we found is it's not easily available. So is it true? Like we try to
Uh,
search many of the databases.
See, multi-omics means that data generated from different types of omics experiment. It could be genomics, transcriptomics, metabolomics, metagenomics, etc. So first of all, you have to find the paper that has reported that data. Then the paper would have given some links or accessions to retrieve that data from the databases in which they have deposited. Or sometimes they also offer it locally on their websites, and you can get the data from their website, right? So it may take some effort sometimes, but the data is generally available, right?
Okay, okay.
Or
For the
if you are
same,
not able to find, then the best way is that you can write to the last author in that paper, who is generally the corresponding author. Okay, their email address is given in the paper, and you can request for the data. And if they don't reply once, you can write more emails also, reminder like that. After I think 2-3 emails, you should be able to get some response from them and the location of it. Okay.
okay, okay, sir. Thank you so much.
Okay.
So just one more question on based on what you had told before.
What is the question?
Yes, sir, you had told that without taking the tissue, like we will be able to like analyze the transcriptome. So is it through liquid biopsy?
No, I'm saying in the future it may happen, right?
Okay, okay.
So
So
yeah,
still technology
okay.
hasn't come.
Yeah. So
Ah,
people are thinking, and I think ultimately, um, without basically the purpose is that using any non-invasive method, you should be able to measure the real-time expression. So someday I think that will also happen, but I'm not aware that of such a thing at present, right? Yeah.
okay, sir. Okay. Thanks.
Okay, so if there are no more questions, we can end the session for today. Okay, so we can end this session now. Okay, next week we will take week 11 and 12 also we can take together, 11 and 12, because I think 12 is also online now. And then you also have exams soon, so it is better to conclude these sessions at the earliest. So the next session may be the last session that where we can cover both week 11 and 12, right? Okay. Okay, so good for today. Thank you all for joining and see you next week. Thank you. Bye-bye.